So so how should Joe just recording now, so.
Okay. All right, so thanks very much everyone for coming to the January edition of the Delaware Valley Mass Spec discussion group. Before we go into this at all, what I want to do is first thank our sponsors and thank them for their patience as we get things back in order. So we're working on some correspondence that is going to make this more official. We want to thank Mobile Ion, Agilent, Bruker, Gentek, IonSense, Geol, Keystone, LECO, New Objective, NEMS, Phoenix S&T, Restec, Psyx, Shimadzu, Adaptis, Thermo, Trajan, and Waters for all of their help. And if you haven't got back on board with the sponsorships that we're having pre-pandemic, that's what we're going for now. So we would appreciate all of your help with that. And before we go any further, I want to confirm that everyone can follow us on LinkedIn and check out the Instagram and YouTube channels. And Lissa has agreed to let us record this talk, so it'll be up on the YouTube channel soon for everyone to see. If you can tell your friends about it if they're not seeing it now. And one more quick thing that I want to talk about here is this Hplc conference that someone brought up to Harsha and my attention. Hplc 2022. They are talking about going back in person for the first time in 3 years in June from the 18th to 23rd. And if you're curious, you can still make ASMS and then go to this. So please check this out. We're going to put something on our website. And we would like very much if everyone could check it out. Lissa, if you want to take over, I'll go. If you want to share your screen, I'll go over your bio and introduction.
Sure.
All right. So it is my pleasure to introduce Lissa Anderson, who's research faculty and the Director of Biological Applications at the National High Magnetic Field Lab, the Mag Lab at Florida State. She was born and raised in Virginia and attended William and Mary, and then did her PhD at UVA under Donald Hunt with Jeff Shabanowitz, or Donald Hunt and Jeff Shabanowitz, who have really kind of pioneered ion-ion reactions, electron-based fragmentation, and all those great sorts of things in that time. In 2015, she moved to Tallahassee, and she became a postdoc in the Mag Lab's ICR program and joined the research faculty the following year. And she works predominantly on top-down proteomics, and this includes development and applications of technologies for ion-ion chemistry, proteoform detection, sensitivity, primary structure determination. She's also been privileged to work with dozens of users and collaborators domestically and abroad, and develop optimized strategy for characterizing proteoforms involved in really interesting phenotypes. She enjoys curating an impressive indoor jungle, which is very cool, and tending to her native pollinator garden. And if you get a chance, I would highly recommend going on the Analytical Chemistry or ACS website and just typing in the words Lissa Anderson if you're interested in top-down proteomics, because the first 2 pages are basically a whole bunch of papers that are really amazing, and I would recommend everybody reading. And with that, I'll pass it over to Lissa. So please take it away, Lissa.
Thanks, Joe, and thank you so much for the wonderful introduction, but also for inviting me to speak at the meeting of the Delaware Valley Mass Spectrometry Discussion Group. The first talk that I ever gave publicly in my life was at the Baltimore area, the Washington-Baltimore area Mass Spectrometry Discussion Group, which is my local representation when I was in Charlottesville. So, these communities of people coming together to stay up to date on the latest in mass spec technologies, but also, sharing and hopefully developing long-term collaborations, or at least networking is of such great value to everyone in the community. So thank you all so much for the invitation. I'm very honored to speak to you today. I just want to confirm that everyone can hear me all right.
Sounds great.
All right, great. Okay, so before I start, I'm just gonna stop, if I can figure out how. I was going to stop using my camera because I find it a little distracting. There we go. All right, so today I'm going to be speaking to you all about strategies for analyzing top intact proteins, which is otherwise known as top-down proteomics, specifically using the equipment that we have at the National High Magnetic Field Laboratory, which includes a magical 21 Tesla FT-ICR mass spectrometer that you see in the picture there. And if my slides would move forward, hold on one second. Come on. Okay, so, we all know that the primary reason that anybody does a mass spectrometry-based proteomics experiment is to learn the extent to which DNA and RNA level variation combined with the multitude number and combination of post-translational modifications explode essentially the complexity of the proteome, which is comprised of proteoforms. And for anyone who's not familiar with the term proteoform, it's a wonderful word that was missing for a long time. And it was originally coined by Lloyd Smith and Neil Kelleher to mean each unique molecular form of a protein product of a single gene. And if you do biological mass spectrometry, specifically proteomics, we're all very aware that the specific proteoforms that are expressed in cells are really tightly linked to their functioning and to the complex phenotypes in which they underlie. And so, the ability to analyze proteoforms is really dependent on an ability to study proteoforms intact as they existed from the systems from which they're derived. Unfortunately, that's a lot easier said than done, especially once we get above around the 30 kilodalton mark. That's where most commercially available mass analyzers sort of top out in their ability to perform isotopically resolved proteoform analyses. And the reason that is really a question of the signal to noise associated with the analysis of intact proteins by mass spectrometry. And that signal to noise is governed chiefly by the charge capacity of your ion trap and the number of unique fragments that you need to create in order to get the sequence information that you desire or the site localization of the modifications that you desire. And I want everyone to note that I said ion trap charge capacity, not just capacity, because the number of ions that your ion trap or any mass analyzer for that matter, well, any ion trapping mass analyzer, the number of ions that it can hold is dictated by, the number of charges that it can hold. And so, in bottom-up proteomics, we're typically working with small precursors that might be 1 to 5 kilodaltons. They might have 3 charges. And if the charge capacity of your mass analyzer is around 1 million charges, as is the case for 2D linear ion traps or for Orbitrap mass analyzers, you're working with around 3E5 total ions. And let's say that peptide that you're interested in sequencing is 12 residues long. And that means that there are 22 unique sequence informative fragments that you're trying to create, that is, fragments that contain the N or the C terminus. And so if you do the math and you pretend that you create all of those bonds, or you break all of those bonds equally in your mass spectrometry experiment and create all of the fragment ions in equal abundance, which is, probably never going to be the case, but you're detecting around 15,000 max of each unique fragment ion. When we extend this to intact proteins, we again have 1 million charge capacity in our ion trap or in our Orbitrap. And instead of working with 3+ precursors, now we're working with precursors that carry 30+ charges. So we're down an order of magnitude in terms of the number of precursor ions that we can start with. And instead of creating, 22 unique sequence informative fragments, as we would for a peptide, we can create up to 304 unique N or C terminal fragment ions, depending on the fragmentation method employed. And so if you do that math, we're down many, many orders of magnitude, not only in the number of precursors that we start with, but also in the number of fragment ions of each unique fragment ion that might be detected. And so in addition to that, all of those precursors and all of the fragment ions exhibit a much wider range of charge states and isotopes, which is also very detrimental to signal to noise. So ultimately, that means that in order to successfully do top-down proteomic type studies, it requires the averaging of considerable numbers of spectra in order to produce a useful sequence informative spectrum. And that's, when it becomes the most challenging, especially when it's performed on an LC timescale. The other thing to keep in mind is that all proteins are different. They're different sizes. They have different gas phase conformations. They fragment differently. And when you're doing an LC-MS experiment, you don't have the opportunity to manually tune and optimize the data acquisition parameters for each protein as they're coming off of the column. And that's what's enumerated in the initial pilot project for the consortium for top-down proteomics. They laid out some of the important challenges that are faced in top-down, and specifically getting reasonable top-down data on timescales that are compatible with chromatography is really a struggle. So as I said, I'm just going to go through some of those challenges regarding signal to noise. When we look at the intact MS1 spectrum of a variety of proteins, as you see here, we see a distribution of charge states, and that charge state distribution is governed by the amino acid composition of the protein, as well as the gas phase conformation. But what you really need to know in denaturing top-down, as is the case when you're doing chromatographic separations on the front end, for the most part, the thing to know is as the size of the protein increases, so does the width of the isotope, the width of the charge state distribution, as well as the mean charge state of the distribution. So if you have like a 9 kilodalton protein like ubiquitin, that exhibits 7 charge states or so. But as you increase in molecular weight, the number of charge states also increases until we get to, the carbonic anhydrase, which is 20 kilodaltons, that is present in more than 20 charge states. And then finally, once you get to 50 kilodaltons or higher, you're looking at easily 40 charge states or more. And that's a really big problem for top-down proteomics, because as the number of charge states increases, the signal intensity of any one individual charge state is lowered. And so as the proteins increase in molecular weight, the width of their charge state distributions increase as well. And the signal to noise of any one charge state isotopic peak cluster is limited significantly by the number of charge states that it exhibits in the gas phase. And so that's what, we're used to looking in proteomics. When you do LC-MS experiments, we're used to looking at base peak chromatograms, which I'm showing you in red on the trace that I'm showing here. So the base peak chromatogram is essentially the trace of the most abundant charge state, or the most abundant thing coming off of your column at any given time. Whereas the total ion current is the sum of all of the ion current from all of the charge states. And that is what I'm showing you in blue. And I just want to point out that this point on the red trace at 1E7 is an order of magnitude lower than this point on the blue trace. And so the discrepancy, or the difference between, the signal intensity of your base peak versus your total ion chromatogram is really significant, especially when you get to the larger proteins in this standard mixture, as I'm showing here, like the bovine carbonic anhydrase or the Clinau fragment, which is 68 kilodaltons, in which you basically almost don't see a very distinguished peak in the base peak chromatogram. In addition to, your precursor charge, or your precursor being split into a variety of charge states, you also have to deal with all of your fragment ion current exhibiting a variety of charge states. And the reason for that is because your precursor, while you're isolating ions of the same M/Z, that does not necessarily mean that they have the same distribution of charge throughout the molecule. So I always sort of show proteins represented by these little rectangle bars. And so if you have two precursors that have the same M/Z but are different in the location of a charge in the gas phase, and they undergo ETD fragmentation at the same place, you're generating fragment ions that have different M/Zs, and therefore you're putting a ceiling on what the signal to noise ratios can ultimately be because these fragment ions exhibit different charge. In addition to this issue of splitting our ion current significantly on the precursor and fragment ion levels, we're trying to fragment these things so that we can sequence them as comprehensively as possible and site localized post-translation modifications. And when you look at spectra of intact proteins, like the one that I'm showing here, which is an ETD-MS spectrum of bovine carbonic anhydrase 2, you can see that the spectrum is very, very dense with many overlapping peaks that are really difficult to resolve and deconvolve and interpret. So in addition to splitting our ion current because of charge states, we're also actually creating a variety of fragment ions, not just the ones that you associate with a particular dissociation technique like C and Z dot ions in the case of ETD. There are also small numbers of A dot and Y ions that I identified in collaboration with Dr. Shabanowitz of the Hunt Lab in the spectrum that I show you here. So while there are only 258 unique cleavages that can occur because of the number of charge states that each fragment ion exhibits, in addition to the variety of different fragment ion types, within that one spectrum alone, we're identifying over 1,200 fragment ion peaks. And that's just to impress upon you all the dramatic complexity of the data derived from intact proteins, which increases exponentially as molecular weight increases as well. Despite that, we're able to get 91% sequence coverage of this protein, which is pretty darn good, considering that it's 31 kilodaltons or 29 kilodaltons, excuse me. Now that spectrum was kind of cheating. It's taken as the sum of 1,500 transients, which is the same as the average of 1,500 scans. And the data are really, what is happening? I repeated, oh, here, this is where I'm trying to go. The data are really complex and rich. And this is just an example of me showing you 8 different fragment ions associated with the cleavage event, like the same backbone cleavage event of the NC alpha bond, but they all exhibit different charge states in this one spectrum, as well as the generation of some low level Y ions associated with those Z ions. And so I just want to go through and show you what a fully annotated intact protein spectrum looks like. And at low M/Z, it doesn't seem like it's anything too scary. There aren't too many fragment ions, but as we increase our M/Z, this again is that carbonic anhydrase spectrum I showed you before. You can see that we have hundreds of fragment ions in the span of 50 M/Z or so, as well as some fragment ions that I didn't expect to see, like some B ions in there mixed with my ETD generated C and Z ions. All this to say that top down is really hard, and there are a lot of limits associated with the signal to noise issues that I've outlined, but also just interpreting the data can be really a difficult challenge because the spectra are so complex. In addition to this issue of widening charge state distributions, we also have to keep in mind the contributions that are played by widening isotope distributions, that is the incorporation of heavy isotopes of carbon, hydrogen, nitrogen, oxygen, and sulfur. And, you know, most of the time when we think about looking at peptides, this is what our isotope distributions look like, something like this, where our monoisotopic peak is the most abundant peak of the distribution. Or maybe if it's not the most abundant peak, it's certainly at least still present, although at lower relative abundance than, say, the A+1 peak. But when you go to intact proteins, once you get to something, the size of ubiquitin, the monoisotopic peak is of really, really low relative abundance. And if you get much bigger than that, unless you know the identity of the protein that you're looking at, you'll have no a way to assign which isotopologues are which, and therefore you won't have any ability to determine what the monoisotopic mass of a protein or a particular isotopic peak cluster is, because it won't be differentiable from the noise because it's of so low relative abundance. So, you know, as the protein's size increases, so does the incorporation of these heavy isotopes. And that means that we have to rely on a lot of tools that are definitely not perfect, like deconvolution, for example. But the point is that it complicates, you know, the mass determination, but also significantly contributes to this decrease in signal to noise, and also increases your demand in resolving power as mass increases. So I'm very fortunate to work where I do at the National High Magnetic Field Laboratory in Tallahassee, Florida. The facility is a user facility that is open to any and all qualified users. And to be qualified, all that means is that your data has to be publishable. It cannot be proprietary. But people from all over the country and from even around the world travel to the Mag Lab to use some of the ICR instruments that we have there. And the reason for that is because there are a lot of important advantages associated with higher field strength in ion cyclotron resonance mass spectrometry. The mass resolving power and the spectral acquisition rate increase linearly with the strength of the magnetic field. But your mass measurement accuracy and your charge capacity, that is the charge capacity of the ICR cell, also improve quadratically with the strength of the magnetic field. And so that's really, really important when you're trying to do top-down proteomics experiments, because we have the signal to noise issues. And so here's a schematic diagram of our instrument. The front of the instrument, which you might be able to recognize from the picture that I'm showing here, the front is actually a Thermo Fisher Scientific Velos Pro dual linear ion trap that has been modified to include the glow discharge reagent ionization source for the creation of reagents for ion-ion reactions, as is implemented on the Fusion and the Eclipse platforms that Thermo has released. So the front is our dual pressure, or our front end ETD enabled Velos Pro dual linear ion trap. And immediately behind that is probably the most important piece of the instrument to me and to top-down proteomics, and that is a multiple storage device, or an MSD. And what this configuration allows us to do, also please note that none of these things are drawn to scale, but having this instrument configuration is really important because it allows us to accumulate our precursor of interest. If we're doing ETD, we can accumulate our reagent in the Velos Pro dual linear ion trap. We can fragment it, and then we can transfer our fragment ions to the multiple storage device and hold them there. And we can repeat this process, which we have termed multiple fills, We can repeat this process as many times as we believe to be appropriate for the sample or the types of proteins that we're analyzing. And then all of those precursor fragment ions can be transferred to the ICR cell, which is back here, for excitation and detection all at once. And that's very important because the charge capacity of the ion trap, which is in the front, is about 10 times higher than the charge capacity, or sorry, I messed that up. The charge capacity of the ion trap is 10 times less than the charge capacity of our multiple storage device or of the ICR cell for that matter. So if we consider, most ion trapping mass analyzers have a charge capacity of 1 million charges. The ICR, the 21 Tesla ICR system has a charge capacity that remains linear in terms of the signal to noise through an order of magnitude higher than that 1E7 in the case of isolated precursors. And then if you're actually fragmenting with ETD, which would destroy charge, you can keep it linear mostly through almost 3E7, which, for the larger proteins in the mixture, which makes sense. So the multiple storage device is very important for bridging the gap between the charge capacity of the front of the instrument and the charge capacity of the actual ICR cell where we're doing our high resolution detection events. And so what does that buy us? Why is that so cool? Well, traditionally, when we're talking about improving signal to noise, we do that by summing transients, or another way to say that is we average spectra. And when we do this, the signal to noise of the spectra, or of the peaks in the spectra will improve by the square root of the number of scans that you average. But for every volume of ions that you analyze, you're also acquiring a transient, which takes up the bulk of the analysis time. When we do multiple fills, and I want to show you what that looks like. So this is summing transients. This is a CIDM-SMS spectrum of myoglobin, which is 17 kilodaltons about. And I've taken this spectrum, which the large spectrum is the full spectrum, the unzoomed in portion. That spectrum was taken as the sum of 16 transients. So by doing those 16 transients, we've improved the signal to noise by a factor of 4. And when we do this, the amount of time that it took to acquire this spectrum is 16 seconds. And 16 seconds is totally an unacceptable length of time to acquire one spectrum, especially if you want to retain chromatographic compatibility. Now, with the instrument and the configuration that we have it with the multiple storage device behind the Velos Pro dual linear ion trap, we can perform multiple fills. And when we perform multiple fills, the signal to noise improves linearly with the number of fills that we perform. So we get a very dramatic improvement in signal intensity as a function of the number of ion volumes that are ultimately set down to the ICR cell for excitation and detection. But we aren't acquiring a transient per ion volume. And so that saves us tons of time while simultaneously dramatically improving signal to noise much faster than could otherwise be achieved. And so that spectrum I showed you before took 16 seconds to acquire. The same spectrum taken with the same same data acquisition parameters, with the exception of it was taken as a single transient or no averaging with 4 fragment ion fills for the same signal to noise improvement. This spectrum's taken in 1.5 seconds instead of 16 seconds. And you can see that the signal to noise in this spectrum is very comparable to the signal to noise that I show you here in the spectrum that takes 16 seconds to acquire. And so the multiple fills is really the most powerful tool, that we have to dramatically improve the signal to noise fast enough that we can retain chromatographic compatibility of the analysis. There are a lot of other problems associated with analysis of intact proteins. As I showed you in the carbonic and hydrase spectrum, there's a lot of signal overlap in the fragment ions. And the reason for that is because when you're working with intact proteins and you have a protein that's say 30 kilodaltons and it has 30 charges, it doesn't really matter if you break it in the middle of the protein or if you break it more on one end of the protein than the other end of the protein. All of your fragment masses, for the most part, are going to be very closely clustered around the precursor M/Z. And so you're going to see a lot of fragment ion overlap. And that really increases the demand in the resolving power capabilities of your analysis. And so this is a picture of what that looks like with a protein that's say 50 kilodaltons. This is protein AG, a recombinant protein. And this ETD spectrum was taken as the sum of 50 transients at a resolving power of 600,000 at 400 M/Z. And the reason that I show this is just to show you how ugly it is and how terrible it is, and to communicate to you that it doesn't matter how much resolving power you can bring to the table or how long you can sit on a protein and average spectra after spectra after spectra, if all of your fragment ion current is sitting on top of itself within a 1000 M/Z window of your analytical range. This is literally the most powerful mass analyzer in the world, and I can barely get any sequence information from a protein this large because of the extent of fragment ion overlap, as well as the complexity of the actual data that you can't even get a glimpse into from these spectra. And so one thing that I really started working on a long time ago when I was a graduate student in the Hunt Lab was the use of ion-ion reactions to sort of combat this spectral congestion problem. And so we use proton transfer reactions, or PTR, which was originally developed by Jim Stevenson and Scott McLuckie in the late 90s, which involves use of radical anions of perfluorinated hydrocarbons, for the most part, that act as Bronsted bases. And they abstract protons from the precursor ions that we're interested in sequencing. And they abstract these protons without causing any dissociation of the backbone or of any side chains. And so all you get as a result of this reaction is a charge-reduced product ion without any dissociation. So the main thing to take away is the products of these reactions are going to have higher M/Zs than the precursors because their charge states are changing. And the rate of the reactions of these gas phase ion-ion reactions is directly proportional to the square of the charge of the precursor. So a precursor that has 50 charges, let's say, will react much more quickly than a precursor that has only, say, 3 charges or 4 charges. And that's really important when you go to apply this to intact protein analysis, because as I showed you before on the left, there's that ETD spectrum of protein AG with 5 milliseconds of ETD. And if we take those fragment ions and we do nothing but subject them to 10 milliseconds of exposure to PTR reagent perfluoromethyldecolon, and they undergo proton transfer reactions, those higher charge fragments are going to be charge reduced faster than the lower charge fragments. And so that sort of allows the fragment ion signal to be concentrated among fewer channels and to become normalized. And as you can see now, once we perform PTR on these fragments, the entirety of the analytical range that we have available to us from below 1000 to 4000 M/Z is occupied with signal that's actually useful to us, that we can identify and use to sequence and site localize post-translational modifications, if that's the goal. And so if you compare the sequence coverage that you get from the ETD only spectrum of this 50 kilodalton protein, you get around 12%, and that's being generous, you know, allowing for deconvolved masses to potentially even match the sequence and error, because there's just so much signal everywhere. False positives are actually really common in these types of spectra. When we do ETD and PTR, these fragments were actually verified manually and confirmed, but with the ETD and PTR, our sequence coverage increases from 12% to 39%, which is really significant when we're talking about sequencing a 50 kilodalton protein. So, you know, the other thing that we can use these proton transfer reactions for, in addition to sort of normalizing the charge state distributions exhibited by fragment ions and using the analytical range that's available to us, is another way that we can use these reactions is to combat this issue of widening charge state distributions as the molecular weight of proteins increases. And the way that we do that is by coupling the proton transfer reactions that I described before with a method called parallel ion parking, which of course was also developed by Scott McLucky, or in the McLucky lab in the early 2000s, along with several of his former graduate students and postdocs. We sort of picked up the mantle on this in the Hunt lab when I was a graduate student, and we first got our glow discharge front-end ionization source for the creation of reagents for PTR reactions. But all of that is detailed in a lot of the, the references that I include below. But in short, what we're doing when we perform PTR with parallel ion parking is we're applying a waveform that's comprised of frequency components that correspond to the M/Z of product ions that we want to save from undergoing further charge reduction reactions. And so we apply this waveform to the X rods of the two-dimensional RF linear ion trap. And so the proteins or the precursors are allowed to undergo charge reduction until they are charge reduced to the point that they've reached that M/Z that feels that supplemental waveform. And once that is felt by the precursor ion, that increases its kinetic energy and its velocity. And the rate of ion-ion reactions is inversely proportional to the cube of the differential velocity between the reacting ion clouds. So the differential velocity between our precursor protein ions and the reagent per fluoromethyldecalan. And so just a small little bit of activation of those M/Z species of interest causes a really significant reduction in the rate of further charge reduction. And that allows us to essentially park, that's where the term comes from, park our ion current into just a handful of charge states instead of a wide distribution of charge states. And that's what this looks like here. This is a charge state distribution for human serum albumin, which is around 66 kilodaltons. This is from a real sample, so it's not recombinant. There are a lot of different adducted forms in this charge state distribution that I'm showing here. But the multiply charged precursor ions are allowed to undergo PTR, and once they are PTR'd, or their charge reduced such that their M/Z is above around 1730 or so, they stop undergoing further charge reduction reactions. And this allows for a 15-fold improvement in the signal to noise of any one charge state isotopic peak cluster over any charge state in the previously normal MS1 spectrum, I should say. And so we recently published these data and these methods in analytical chemistry. Me and my co-authors, I especially want to call out my co-author, Chad Weisbrod, who is also my colleague and friend. And we couldn't have done any of these things without him, but we're very excited to have PTR capabilities on both our 21 Tesla and 14.5 Tesla instruments, because we, have seen and will continue to see routine improvement in signal to noise by a factor of say 10 or more, depending on the molecular weight of the precursor ions that we're interested in. In fact, here are a few other examples of improvements that PTR and parallel ion parking can bring to the table. So this is a spectrum of a recombinant mixture of all of the different isoforms of tau. And I'm showing the chromatogram here. I was doing these experiments for Wilfredo Russell of the Mayo Clinic of Florida, and I had never run tau before. And it's really unfortunate that despite the fact that there are, several different forms of tau that span around 36 to 46 kilodaltons, they all colute within the same 2-minute window, of course, as is always the way. And so if you zoom in on any, like on this portion of the spectrum that you see here, you can sort of see isotope distribution starting to appear. This is not very much averaging that I have the opportunity to do. And you could determine what the charge state is if you, did the math in a very crude way, but you certainly wouldn't be capable of determining the monoisotopic mass of any of these species. When we take these and we perform proton transfer reactions with parallel ion parking, we get this spectrum that I'm showing here with a really beautiful distribution of all of the different isoforms of tau that are present in the sample, all detected with sub ppm mass measurement accuracy at 21T. And so that's just another example of the power, of when you have so many different things co-eluting at one time. A lot of times from the regular denaturing ESI MS1 spectrum, you wouldn't be able to get very good data for an intact mass determination, but with PTR and parallel ion parking, that's easily gotten. We also performed an experiment with a very complex sample that was prepared by Dr. Leah Schaefer, who was at the University of Wisconsin at the time, but has now moved on to her postdoc, I believe in California. But we performed LCMS with PTR and parallel parking, and without PTR and parallel ion parking on some gel-free fractions of an MCF7 cell lysate, which is a breast cancer cell lysate. And we wanted to compare the number of proteoforms that we detected with and without PTR and parallel ion parking. And so this is what some of those data look like. On the left, you have the regular MS1 spectrum without PTR and parallel ion parking. And on the right, you have the PTR PIP spectrum with the region in gray depicting where the parking waveform is being applied. And so if you look at the spectra here, the signal to noise improvement is almost a factor of 5 over the regular MS1 data. And then very importantly, if you look at the raw data, this is the regular MS1 data, and this is the PTR PIP data. If you take the raw data and you subject it to deconvolution with an algorithm like extract or thrash, that's ultimately what's going to determine, how many proteoforms are identified in these data, because you can't identify a proteoform without an intact mass match, at least with most, data analysis platforms that are out there anyway. And so when you look at the metadata derived from the raw data following deconvolution, we see that there are way more proteoforms represented in the PTR PIP data that's ultimately used for identifications compared to the deconvolved data derived from the regular MS1 data. And so ultimately, that translated to the detection of more than 1,000 more proteoforms in the PTR MS1 mode data compared to regular MS1 mode data, with many of those being higher molecular weight proteoforms compared to the regular MS1 mode data. Another thing that we need to think about, and when we're doing top-down proteomics, we're doing data-dependent analyses. And so in addition to improving signal-to-noise ratios, we're also improving ion accumulation times, which is really very important so that we can get as many MS2 spectra as possible. You know, when you're doing a bottom-up experiment and you're looking at peptides there, it's kind of it doesn't always happen that you have a difficult time accumulating the number of ions that you're asking for. But for intact proteins, because there's not a lot of signal concentrated into any one charge state, the ion accumulation times are much, much higher than what people are used to in bottom-up data or in bottom-up proteomics experiments. And so we can accumulate these ions following PTR and parallel ion parking. We can accumulate the same exact quality MS2 spectrum, but we use 33 times less ion accumulation time compared to normal MS1 mode. And that ultimately will translates to more MS2 spectra acquired of more precursors, and therefore hopefully more proteoforms identified. Hey, Joe, how am I on time?
Let's say wrap it up in 5 or 7 minutes. Does that work?
Perfect, okay. Sorry, I had to blow my nose. Okay, so I just want to make sure that I'm sticking on time and I know what I have left to do. Yes, okay. So here's an example of how we can implement a lot of stuff that we've developed at the Mag Lab and actually solve a real biological question with top-down proteomics. That involves analyses of samples derived from patients that have monoclonal gammopathy or multiple myeloma. Multiple myeloma is a cancer of your blood cells in your bone marrow, or your plasma cells, that is. And these are the cells that create and disperse antibodies into your serum for circulation so that they can, keep you from getting bad colds and diseases and COVID and things like that. So normally your plasma cells will, like one clonal population of plasma cells will produce one antibody, and that's always the case. But if you have a cancer like multiple myeloma, the transformed clonal population of plasma cells will overexpress whatever antibody it is that they've been programmed to secrete into your blood. And the concentration of antibody, which in this case is called M protein, in your blood goes up really, really high, and it causes a lot of problems with your blood and with your kidneys and liver and all sorts of issues. But the main takeaway is that these transformed plasma cells overexpress one antibody. And so how normally this is diagnosed in the clinical laboratory is through electrophoresis and Western blots, in which they're looking for this M protein spike, which means that there's a lot of IgG or a certain particular antibody in your blood. And then if they see that M protein spike, they will perform isotyping to figure out what type of IgG is it. Is it an IgG1 or an IgG2, et cetera? What type of light chain does it have? Things of that nature. But the issue is that these clinical assays have high detection limits. and low specificity. So you don't learn anything at all about what the antibody sequence is. You only learn what its isotype is and therefore what its constant region sequence is. And so this is what all the IgGs look like purified from your plasma cells. And if you were a normal, healthy individual, you would sort of have this Gaussian distribution of a polyclonal background that you can't really take anything away from. But if you have monoclonal gammopathy or multiple myeloma, the signals associated with that one overexpressed antibody are going to be very dominant. And so this is what the data looks like there. And so one question that a lot of people have are, people undergo this relapse and remission cycle often, where the concentration of M protein in their blood increases to a point where they become symptomatic. And then they undergo a therapy and it plummets. But then during this time, this plateau or remission time, nobody has any understanding of what's going on with these antibodies, because they're above or they're below the detection limits of the standard clinical assays. And so these assays suffer from several limitations, including this lack of specificity, but also they're, painful for a patient because it involves a bone marrow aspiration and they're expensive. So we set out, and by we, I mean, I did this in collaboration with folks at the Mayo Clinic in Rochester, but also a former student of ours. His name was Li Dong He. I have to show you his picture because he's amazing. So that's Li Dong, and he was a graduate student at the time that we were spearheading these experiments, and he did a really terrific job. So we kind of wanted to know what was going on at this time. And so, with the top-down stuff, we can get tons of sequence information with PTR and parallel ion parking. In just a single experiment using an average of 10 transients, we can get 74% sequence coverage. Then if we do a separate experiment, we can get 85. But we also were working on, this is an old slide, bad Lissa. This method, which allowed us to do ETD and CID in the same experiment and look at those fragments in the same spectrum. That's called chimeric ion loading. We can use that to really quickly sequence antibodies much faster than performing two separate experiments. And that just involves using our multiple storage device to perform CID fragmentation, store those fragments, perform ETD fragmentation, store those fragments, and then send them to the ICR cell. And so that's what this looks like. The CID spectrum is on top with ETD on the bottom, and you can see in our chimeric spectrum, we have all of those fragments in a single spectrum. I'm going to go ahead and skip this because I know we're close on time. But essentially, this is the light chain of the antibody that I was showing you from a patient with multiple myeloma. And because of all of the sequence information that we're able to glean very quickly, we are able to fully sequence the antibody based on the genome sequencing data. 68% of the antibody was covered, and 100% of the assignments that we made matched the genome sequencing data, including 7 different single amino acid or single nucleotide polymorphisms that resulted in single amino acid substitutions. And so this is really a nifty method because you don't have to have a bone marrow aspiration. All this is done with just about 20 microliters of serum. And the detection limit's about 10 times lower. But in addition to learning the isotype, we can actually determine what germline sequences these antibodies are derived from. And that allows the patients to be stratified for personalized treatment. For example, like one isotype might hint more at liver involvement in the future than other isotypes. So the doctors and clinicians like to have that information so they know what treatments are best for their patients. In addition, because we can do this at such low levels and we get so much sequence information, we can monitor the disease progression and the cell mutation status in ways that have never been monitored before. And so I know I've gone over time, and I want to just go ahead and close there and thank you all for your attention, as well as point out, you know, the great people that support the Mag Lab at the NSF, the Division of Materials Research and the Division of Chemistry. And I want to thank our collaborators on PTR and parallel ion parking, the Hunt Laboratory, and the folks at Thermo Fisher Scientific in San Jose. So with that, I'm happy to take questions, and I apologize for going over time. And also, I'm sorry, I'm losing my voice.
All right, thanks very much, Lisa. That was amazing stuff, and I especially thought it was an amazing application at the end that showed off all of the cool work that it could do. So we do have some time for questions, and as the chair, I get to go first, so I'm going to take full advantage. So one of the things that is so cool, and I used to work on top-down a lot, and we would find the same thing that you found. No matter what resolution you have, if you can't spread out the fragments, it doesn't help any. So one of the things that we always thought about was doing ion mobility separation post-fragmentation. And this was something that we wanted to do, but there's only one instrument that could do it, and it was the Waters Synapse, which doesn't really have the resolution that we wanted. I was wondering if you could kind of pontificate on how parallel ion parking compares to a technique like that, that's an actual gas phase separation, and which one you think would be better, or if there are plans to do that in the Mag Lab. I think you're on mute right now, by the way.
We have a lot of interest in ion mobility, and we've looked into putting an ion mobility instrument on the front of the ICR system, and it would really open the door to some very interesting applications that can't be done anywhere else, that's for sure. Forgive me, I need to blow my nose, I'm so sorry. So in terms of like the ion mobility stuff that Christian Bleiholder is doing with the TIMS TOF, that is like this, that's the way that I would want my ion mobility system set up, because then the coolest thing about it would be the ability to isolate precursors of a specific mobility so that you can fragment them separately from other precursors and then look into differences in gas phase structure, or perhaps different post-translationally modified forms have different gas phase structures. And that would be a way to get sort of a cleaner precursor ion population compared to what you're able to get with regular old waveform isolation, where you're isolating. I mean, I would imagine the spectra would be so much cleaner if we had ion mobility based precursor selection, because you wouldn't have mixtures of precursors of potentially greater variety of charge or different localizations of the same PTM that gives it the same precursor M over Z, but different fragment ions. So I love ion mobility for that reason. The thing that makes it difficult to put on the 21T is that we have our own little time of flight effect. And also because, I mean, there are a lot of different reasons, but that's the primary one, is just that you kind of need to figure out a way to trap the ions separately externally to that, which we could do. I should talk to them more about, we have so many fun things we want to do, you know?
And I'll ask one more question, because I think it's going to be quick. I know there are a lot of people chomping at the bit. So one of the things that I always think about is how ions are somewhat considered metastable once they're in a mass spectrometer. If you could think about the stability that you have with the manipulations you're doing in your multiple storage device versus the stability that we have in, for example, an LTQ, you know, what's the difference? Is the MSD, are you allowed to get away with a lot more without losing anything than you would in an ion trap, or are they pretty similar?
They're pretty similar, especially for things that are the same size. Like, so if you're doing a lot of MSMS and you're looking mostly at fragment ions, you can kind of operate it exactly the same way. But there are certainly differences in the way that we operate it for small molecules versus intact protein precursors. We have to think about the frequency of that we drive the multiples at, the arrival time, the time of flight that it takes for the ions to get down, and what their M/Z's are. So like at higher M/Z, their arrival times are going to be a lot later than stuff at lower M/Z. So being able to modulate things like that in the multiple storage device independently of a lot of other stuff will become more important as we try to do fancier and fancier experiments, or try to simultaneously trap, something at 20,000 M/Z, but detect a fragment ion down at 300 M/Z. So stuff like that. I hope that answers your question.
Absolutely, yeah. All right, I'll turn it over to the rest of the audience here. So please feel free to ask Lisa questions if you have any. I know Harsha has some.
Lisa, wonderful talk. Really enjoyed the talk. You were showing this parallel lab parking. I was in the lab that did that 15 years ago. It's called Hplc's lab, and we did parallel parking around the same time. I was in the lab. So it's interesting to see that you put this on an ICR. I was just wondering if you were using filtered noise fields on the linear ion trap to do this, or was it in the storage device? I wasn't clear where you were doing the ion parking. Is it in the linear ion trap or in the rest of the instruments?
It's in the ion trap because you have to be able to do charge sign independent trapping. So we have to do the ion-ion reactions in the ion trap. Okay. So that we can simultaneously have the negative reagent ions and the cations. If we designed a different multiple storage device that was segmented in the way that the linear ion trap is from thermo, we could do them elsewhere. There's no reason that we couldn't do some experiments in the multiple storage device without, with kind of a minimal amount of changes to the hardware, but the ion trap definitely gives you the ability to manipulate the ions the best way.
So you were using filtered noise fields to do this?
We're using what?
Filtered noise fields, so you were using multiple waveforms, right, to do parallel?
Yeah.
To trap ions in parallel.
Yeah.
But you're doing single, but you're fragmenting up, you're doing ETD or whatever fragmentation, subsequent fragmentation, once you parked on a single charge state, right?
Yeah, we can do data dependent selection of the PTR park precursors and then fragment them. Yeah,
so is it one precursor or multiple precursors? Because I was wondering if you could park and concentrate, single resonance frequency parking would be better than parallel, because you would pretty much concentrate all the charge states into a single charge state before you fragment. I was wondering why you would select a parallel approach rather than using a single resonance frequency to enrich for one charge state.
That's a good question, and the reasons are twofold. The first reason is because in order to effectively use single frequency resonant excitation, you need to know what M/Z you expect your product to be parked at. And when you're doing an LCMS experiment on a complex mixture of proteins, you don't have any idea what M/Zs those are going to exhibit. So you can't do single frequency parking experiments on the complex mixture stuff, because you can't anticipate what your waveform should look like that you use for parking. The second reason is because just because you apply a waveform doesn't mean that the reactions are stopped altogether. They still do undergo a small amount of charge reduction at a dramatically lower rate. And so by using the waveform with multiple frequencies, you can slow additional subsequent reactions and keep your product ions from reacting so much that they fall outside of your M/Z range compared to the single frequency parking experiments. They would work for stuff like intact protein standards and things that you can anticipate or predict what where you want to park them, but when you're doing unknowns, you have no idea.
Okay, and I was also thinking like, you were showing ETD as a way to dissociate, but there's also a renewed interest in electron capture dissociation using traps, electron activated dissociation, and so forth. So I was wondering, the ICR community, had electron capture before electron transfer. So I was wondering why you were resorting back to, instead of doing ECD, why you were thinking of an ETD approach rather than just doing ECD and just using proton transfer chemistry.
So at the time that we built the 21T was built, there wasn't a robust ECD implementation that you could do outside of the ICR cell. Now there is, but at the time there was not. And the amount of time that it takes to accumulate the number of ions that you need, electrons that you need to do your ECD experiments is kind of high still, even, though it's 2021. So it's kind of difficult to turn on and off that filament that's creating your electrons fast enough that you would get, unaffected MS1 spectra and then MS2 spectra that have your fragment ions of interest. It's sort of just about being able to switch back and forth very quickly as you would normally in an MS experiment. I think even the commercially available one that the last time I checked that one, it was like something where you had to do a run with the filament on. And so all of your spectra were ETD spectra, and then they would do a run with the filament off, and that would be your intact mass run. So for us, we chose ETD because we had big plans, and we also knew that we were going in the direction of wanting to have the proton transfer reactions and parallel ion parking, and the reagent ionization source that you use to ionize ETD reagents. That I was working on that in the Hunt lab before I came to the MAG lab to ionize IIPT or proton transfer reaction reagents. So that's why we decided to go with ETD at that time.
Thank you.
Yep.
So can I ask one more question? It's very interesting to know that you were able to detect variants like mutant SNPs, single nucleotide polymorphisms in a very complicated mixture on the IgGs. Were you able to quantify the amounts, or was it more like you detect them, but were you able to chromatographically resolve these IgGs?
So the ones that I showed you they're not really, like they're so overexpressed that they're the only antibodies that you're going to detect top down. And so it's not really, to us, it was never a question of being able to quantify one relative to other forms, because we only see the one form in these types of samples. We did do a standard calibration curve to demonstrate in one of our papers that our limit of detection for M proteins was 10 times lower than the clinical assay. But I don't know that that necessarily translates to getting the same quality of sequence information. That's just, do you have an M protein in your CRM or do you not?
Yeah, I'm specifically asking about the variants, right? So the polymorphisms, so you said you detected number of different variants for different patient-specific variations. In that case, are you just getting a mass?
I can't really hear him. Can you?
We lost some audio there, Harsha.
Can you hear me now?
Not really.
But remember, these are it's not like we're detecting low-level variants. It's that all of the antibody that's expressed by that transformed clonal population has those variant amino acids.
I see, it's 100%. I see. Okay. So a single patient might have that.
I can't hear him.
Yeah, sorry, Harsha. I think we're coming a little late.
All right.
Is there anyone else that has?
Can
any
I
questions?
have just a simple question, a naive question, because you know that FT-ICR is very powerful, and I'm just wondering, have you ever tried to determine the native protein quantitation? Because the reason I'm asking is that when you have denatured proteins, the number of charge states is too many, and it's complicated for your mass spec analysis. But if you try to use the native condition, you may have a fewer number of the charge, right? So is it helpful for you, for the data interpretation or quantitation? I know there will be a trade-off because your sensitivity only good enough is compared to the denatured protein, but you have a fewer number of charge.
So running these things in denature mode, in native mode, or doing native electrospray would definitely cut down on the width of the charge state distributions of the proteins. The issue is that native proteins don't fragment as well as denatured proteins. And so the sequence information that you can get from fragmenting the native protein is not as good. The second issue is that we're also coupling all of the data that I showed you are with front-end online reverse phase separations. And I find that the reverse phase separations are still the most robust way to separate proteoforms compared to ion exchange type separations that you would perform online. And there would also be a decrease in the sensitivity of the electrospray if you went from native or denaturing mode to native mode. But primarily, I would say that the goal of a lot of the experiments I do is to get the most sequence information possible. And that does not go hand in hand with performing native electrospray ionization. And so, but you're absolutely right. If you do the native analysis, you would have higher signal to noise because you have fewer charge states.
Okay, so the other thing is that because you did not discuss a lot about the LC condition, because I'm just wondering how can we improve the signal by using more separation from the LC side instead of using the mass spec, because you said that there is a limitation of the protein detection in your sample.
Better separations are the #1 thing that would make top-down better.
Okay.
In the context of complex samples like whole cell lysates, having the best separation possible is definitely the #1, I think, way to ensure the success of your experiment, because it's so important that that the fewest number of proteoforms possible co-elute at one time for signal to noise reasons.
Okay, thank you.
You're welcome.
I have a question if there's time. So when you're talking about multiple fills earlier, and how if you do multiple fills, that cuts down a lot on the time of like collecting a spectrum compared to if you did like a bunch of scans and then average them.
Yep.
Is there a point at which you, depending on the charge state or the size of the protein that you're looking at, that you could reach the charge capacity, but still just not be able to achieve, even though the time is shortened, it doesn't matter. You have to resort to going back to averages.
Yeah, I still definitely do, like, depending on the size of the protein, I like to incorporate a bit of averaging in addition to performing multiple fills, not just for the signal to noise improvement, but also for improvements in the look of the isotope distribution, if you know what I mean. For deconvolution purposes, you need that isotope distribution to have relative abundances that are as you would expect them to be in nature. And so I do a little bit of averaging a lot for that reason. There's also definitely a point at which you hit your accumulation time maximum, and then you only have the opportunity to average if you're collecting more scan. It depends on the way that the method is set up, but averaging I still incorporate it into my analyses for sure. And can you remind me of your question though?
Oh, so I was just wondering if like, if you had something that was in a really high, it was in a high charge state and it was a large protein, you showed that figure where it distributes into so many channels whenever you fragment. Yeah. So I was wondering, is there a point at which like, you're gonna reach the charge limit, but it still isn't good enough, like to do multiple fills? Like you won't get good enough data. You'll have to end up just averaging.
Yeah, absolutely. I mean, once you've hit the charge capacity of the ICR cell, you're done. Like there's nothing more that can be gained from doing multiple fills, at which point you would want to be averaging to improve the signal to noise ratios for sure.
Okay. And then one other
but the other thing that I want to point out is we did do some experiments where we showed that like we looked at the amount of sequence coverage that we got for proteins of different molecular weights as a function of the number of fills. And eventually that's going to plateau, right? Like eventually you won't need any more ions and the sequence coverage is just going to be what it's going to be. And having more ions isn't going to help you. In that relationship, we sort of define that as 25 charges per kilodalton of protein. So if you have a precursor that's 10 kilodaltons, we want a cumulative ion target of 1 million charges so that we can get the most we can out of using the multiple fills. And then beyond that, it doesn't make any sense to perform more multiple fills.
Got it.
So you might as well be acquiring more spectra.
Got it.
And
that makes sense.
25 per kilodalton.
And then other quick question is, after you do ion parking, where you can kind of concentrate the charge into certain, certain higher M/Z region of the spectrum, I suppose that doing ETD after that is not the most desirable, just because now you're dealing with a charge state that is, less amenable to ETD. Have you thought about doing UVPD after doing ion parking?
Yeah, we have UVPD capabilities on the instrument, and we can certainly do UVPD. The issue that the reason that I usually don't employ UVPD with chromatography is because instead of creating, 2 or 4 different types of ions, it creates 6 or 9 different types of ions.
Is this the argon fluoride or the ND YAG laser?
There's a well, I guess I don't know.
I don't.
Lisa, is this the YAG laser or the excimer?
Well, we have the Exastar laser, argon fluoride.
Okay.
But I don't think it would. You have a little bit more control, I guess, with the other laser based on what I've seen, but I don't have that system set up in my lab, so I don't have any.
Well, I was just going to say the 193 is way better than the 213.
Is it? Why?
In my experience, there's just way more power.
There is, but I guess I would say also that the spectra do look more complex. Not that the FT can handle that, I think, but if you look at like the 213 data, it's generally a little bit cleaner looking.
Yeah, because it doesn't fragment as much.
Well,
that's why.
I mean, if you hammered away at it, you could. I mean, yes, I'll agree with you that the 193 is going to like blast things away, but either way, I wasn't sure if you had thought about doing 193 with the ion parking.
Certainly, if we had the right sample for it, like the clean sample, and it were a targeted type of method, I would totally consider doing that experiment. But for the like high throughput top down type experiments that are discovery experiments, I'm usually doing CID fragmentation, because it's enough that you get good sequence coverage of the protein without blasting it into 1 million pieces, so the signal to noise is still high enough.
Yeah, that's for sure a definite disadvantage of UVPD is because there's so many channels. It it makes your signal to noise problem even worse.
Absolutely.
So, yeah.
And also the false discovery rate. Somebody needs to do a study about this, but what is false discovery rate when you're trying to match 6 different types of fragment ions or 9 different types versus 2 or 4?
We actually did that. If you check out the paper in 2014 on top-down proteomics with UVPD, we talk about 9 versus 4. And the idea was you just do it with 4 if you can, and you get much better data than if you look for 9.
Yeah, I think the false discovery rate gets like it skyrockets. It gets pretty
explodes.
high.
Yeah.
Right.
But guys, I think with that, I think we're running out of time. Let's all virtually thank Lissa Anderson for providing us with this wonderful talk. And I'm sure that there are tons more questions, and I wouldn't be surprised, Lissa, if you got a whole lot of emails about this talk.
Anderson@magnet.fsu.edu.
Yeah. All right. Thank you again, Lissa, and thanks everyone. And
My pleasure.
have a good evening.
Thank you for the invitation.
Thank you.
All right. Bye-bye, everyone.
Bye.
Thank you.
Bye.